Hello dear all beautiful learners, welcome all once again for connecting with a new class, and we were in the chapter 8 in very last class, and we studied a very important topics in last class, that is the monocot stem, internal structure of the monocot stem. So my dear students, we are near about with a very few topics is left that we are connecting here and are just going to be end of this chapter. Today we will discuss about the identifying features that was not discussed yesterday of the monocot stem, and then we will discuss about maybe in our later I have maybe another one, maximum two class, maybe one class, so our chapter 8 will be completed. So today important topics of the monocot stem, characteristics of the monocot stem, we will try to understand this one. Identifying characteristics. It is also asking for how can you identify a monocot stem? Identifying characteristics. Identifying characteristics of monocot stem. Monocot stem. Yesterday we learned many of the things from the monocot stem, their structure. We draw the figure also, and we just connecting the parts, the epidermal layer, hypodermal layer, and endodermal, that means the lower part, vascular bundle, epidermal tissue, ground tissue, and vascular tissue to discuss also. So and now why and how we can findings the identifying features of the monocot stem. Very first one, epidermis with thick cuticle stem hairs. Epidermis with thick cuticles. Epidermis with thick cuticles, we can see. Can you can think about that, sir? All the times we can it getting that one, that thick cuticles? No, many of the stem, not only monocot, it can be thick, but it is also counted as the identifying features. Okay, I have seen instantly Sabrina Hussain, Nujhat Tasneem, Adhora, all of you, walaikum salam, my dear students, my dear beloved students. Actually we are getting missing that very much time we cannot get face to face. I cannot see that your connection instantly in the study in face to face. By the way, hope so you are joining there, and I hope so I got it here, your presence, that make me very good feelings in them. Okay, by the way, epidermis with thick cuticles, it is the first identifying characteristics of monocot stem we can finding here for getting that one, first one. And very another important, stem hairs are absent. It is very important. Stem hairs absent. It is, though the epidermis with thick cuticles can have founding in different kinds of stem, but stem hairs absent is very prominent identifying features of the monocot stem. It is also very important one features. Cortex is heterogeneous. Okay, very important. Another features is cortex is heterogeneous. Cortex is heterogeneous. Heterogeneous means, actually, homogeneous is similar kinds, but heterogeneous is different kinds of size of the cortical region. The parts inside the cortical region is not similarly, not having the same size, not doing the same activities, not having as like as the previous or any kinds of stems. So it is the heterogeneous. So cortex is heterogeneous is another important features of them. And then later on we will go through hypodermis is sclerenchymatous. That means hypodermis also we know, getting here, hypodermis is sclerenchymatous. Sclerenchyma, that means the hypodermis composed of the tissues of sclerenchyma. So it is also identifying important features of them. And then xylem elements and rich Okay, that means and dark are and dark. Xylem elements are and dark. Very, very important this one. Xylem elements is and dark. And dark. I told you the protoxylem to center, metaxylem to periphery. Protoxylem to center, metaxylem to periphery. So they are called and dark is the typical features for having understanding. If it is asking what is and dark, so on that time it is we will tell about that one. Jui Chaudhuri, alaikum salam. So we have seen there what type of identifying features we can easily identify. It is you are in the intermediate level. So when you will do the practical work in the practical exam, that times after section you can see the under the microscope the important and prominent part and finding the features and tell that what kind of a stem we are using there. So these type of prominent identifying features need to be remembered. My dear students, later on we will go through there. Vascular bundle are conjoined, collateral and closed. Vascular bundle, I wrote the features also yesterday. Bundles are conjoined, collateral and closed. Collateral and closed. Conjoined, collateral and closed, and there is no cambium. That means cambium absent. It is also a very important features of getting that one, that conjoined, collateral and closed and cambium absent. Vascular bundle, I told you also yesterday, before last class, the 27 August maybe that class on going on there. Very, very important topics I discussed about the vascular bundle, their types, with drawing the figure for make you understand easily. So who are not getting that class connectingly properly, so please visit that one and directly try to connect from there. And if you do that class, any kinds of vascular bundle types, their matters, you can easily understand and you can easily remember that one. So vascular bundle are conjoined, collateral, closed, and their another prominent feature is cambium absent. Since the vascular bundle types we discussed there, then we can go through. Yes, it is also this vascular bundle, about the vascular bundle, this one are conjoined, first one, and second one we can write about there. These vascular bundle are numerous numbers, and scattered to the ground tissue, That means we know that the vascular bundle in the center and very lower endodermis types, then after going on the hypodermis and this endodermis, the cortical region, that is counted as the ground tissue. So numerous number of vascular bundle, that is not only for endodermal layer, it can scattered into the ground tissue also. Okay, so it is also very important identifying features for finding that one. Later on we can try to understand. Yes, very importantly we can say Xylems are, we also write in very last class, V or Y shaped. Xylems are V or Y shaped. What the figure can you remember yesterday? Y shaped and phloem cavity found. Phloem cavity found. Of course, there Xylems are V or Y shaped and phloem cavity found there. Of course, this one is very important also. And then we can think about that. What is the later features? Yes, phloem between the arms of Xylem elements. Identifying features. Also, I have written yesterday. Phloem present in between Xylem elements. Xylem elements. Xylem elements. And phloem parenchyma is absent. If we try to find out the phloem parenchyma, that is absent. Phloem parenchyma absent. These are the very important identifying features of monocot. It is asking for several times. It is asking for the identifying characteristics of the above stem. Can you remember that one? So if it is asking on that way, what will we learn? And I will just make you understand in which way. I have seen Aisha also joining, Jaima, Walaikum Assalam, all of you, and your presence is very good and make me thrill as a teacher, because I told you, I not actually, we all, everyone miss each other for connecting in the class and for visible seeing. Okay, by the way, identifying characteristics of the monocot stem is very, very important. That was just lacking part of that previous study or previous class. So what are the features, identifying features is needed to remember, and what are the identifying features is needed to collect properly for understanding this one. Epidermis with thick cuticles. Epidermis with thick cuticles. Though I tell you these features can be any kinds of stem or any kinds of plant root or outer layer, but also have the identifying features counting here. Stem hairs absent, it is very prominent. If you see by physically, stem hair, no stem hair, so monocot stem, maybe instantly we can think about that. Cortex heterogeneous, I tell you the cortical region is not getting in properly. Similar shape and size, all the cortical matters cannot have their similar functional activities, or they are not connecting in similar way. So that is the heterogeneous. Hypodermis is sclerenchymatous. Hypodermis is known as the sclerenchymatous. That means hypodermis is composed of sclerenchyma tissue. And Xylem it is also, we know these features also. Xylem elements is andark. Andark is counted. Why it is counted as the andark? Because protoxylem present to the center and metaxylem present to the periphery. Yesterday when we draw the figure on going on there, protoxylem counted to the present in the center, and metaxylem present to the periphery, center and periphery, the circumference, their presence on going on there. Vascular bundle are conjoined, collateral and closed is counting as there. Conjoined, collateral and closed, where cambium is absent. Okay, vascular bundles are conjoined, collateral and closed. Since they are counting as they are conjoined, collateral and closed, cambium is absent there. It is very important. Another features, vascular bundles are conjoined, collateral and closed, where cambium is absent. Okay, importantly going on there. Numerous numbers and scattered to the ground tissues. Vascular bundle, what we think about that, my dear students, think the figure, what the figure we have drawn there, that vascular bundle are conjoined, collateral and closed, cambium absent on there. So numerous number, their number are very, when you find out that identifying features on that case, so the numerous numbers is counted as and scattered to the ground tissues. That means the numerous number can counted about the ground tissue systems and scattered to the ground tissue, not only for endodermal layer, it is going on through scattered to the ground tissue systems. Xylems are V and Y shaped. We are getting also have features. Yesterday I also write that features the V or Y shaped and phloem cavity is found. That is very important features when you section and can finding below the microscope. What will you see? The V or Y shaped is going on there, and phloem cavity is also getting found from that specific part. And phloem present in between the xylem elements, I told you that the protoxylem and metaxylem are counting as the important vascular bundle for having a connection with that one in between the xylem elements. Phloem parenchyma absent. Of course, this one is also very important. Phloem parenchyma, if we try to find out about the phloem parenchyma, that is absent in this case. So these are the identifying characteristics of monocot stem. So epidermis with thick cuticles, stem here is absent, cortex is heterogeneous, hypodermis is sclerenchymatous, xylem elements is andark, very important, okay, for medical admission test also. Protoxylem to the center, metaxylem to the periphery. Vascular bundles are conjoined, collateral and closed, where cambium is absent. Numerous numbers vascular bundle will find out there and scattered to the ground tissues. And xylems are V or Y shaped, and phloem cavity is found there, and phloem present in between the xylem elements. In between xylem elements, the phloem is also present there, and phloem parenchyma is absent. In this way, total monocot stem we learn. Monocot root is left one topics that we will discuss in the next class. Then we can complete our chapter. So hope so, my dear student, you understand the features, you understand the today's lecture as like as the previous time, and we are connecting well as like as the previous time, and we hope so that we will be better for understanding in the next class. So that's all about today. Everyone, stay safe, stay sound.